Bismillahirrahmanirrahim. This is the June 24 paper 2-1 and this is the second video which has question 4, 5 and 6. Question number 4. Some plants can survive extreme dehydration for a long time in a dormant state. They start growing again when water is available. They are adapted to survive in deserts. Selaginella lepidophylla is an example of this type of plant. Figure 4.1 shows a S. lepidophylla plant after several months without water and the same plant 3 days after rainfall. Several months without water, 3 days after rainfall, you can see the difference. And this is the plant. Plant is a brown color, plant is a green color. Explain how the changes in the plant shown in figure 4.1 will help it to start growing again after rainfall. Well, because basically chlorophyll, the large surface area after rainfall. So photosynthesis is going to take place, glucose is going to be made, or you can say carbohydrate is going to be made, and it's going to trap, or you can say absorb, light energy, because what did it say, it's become green in color. So the fact that it's green in color means that now the chlorophyll or the chloroplasts have developed, and it will be many chloroplasts in it, so there's going to be a large surface area. And of course you can see the leaves also, after rainfall there's a large surface area, so this is going to photosynthesize, and glucose is going to be made, because chlorophyll can absorb, or you can say trap, light energy, and has converted to chemical energy, which is the glucose molecule. So chlorophyll, chloroplast, large surface area, because you see this was all folded and now the leaves have sort of spread out, so it's got a large surface area exposed to light, which can photosynthesize, glucose can be produced, and chlorophyll is going to absorb or trap light energy. Then we come on to part 2. Figure 4.2 shows the cell wall and nucleus of a cell from this plant after several months without water. Suggest and explain the appearance of other parts of this cell after several months without water. You may draw on figure 4.2 to help your answer. So basically, if the plant is not going to have any water, what is going to happen? Loss of water molecules by osmosis, because there's going to be diffusion and evaporation of water vapors from the leaves. So cytoplasm is going to shrink, the sap vacuole will shrink. So you can see some sort of a thing like this, a structure like this, because you see plasmolysis is going to take place. So cytoplasm shrinks, sap vacuole shrinks, cell membranes pulls away from the cell wall. If the cell membrane pulls away from the cell wall, and the sap vacuole has shrunk, which you can see here, the sap vacuole has shrunk. Cell membranes pulls away from the cell wall. This is going to be lack of turgor pressure, and this is called what? This is called plasmolysis. So if you wanted to label it on the diagram, you could have done that, or if you just wanted to write it, you could have done that as well. So basically, you've got to explain it and write it correctly. Loss of water molecules by osmosis, and cytoplasm is going to shrink. This is, of course, we're talking about wilting. Cytoplasm shrinks, sap vacuole will shrink, cell membrane pulls away from the cell wall, lack of turgor pressure, and the word is plasmolysis for this. Then B part of the question, Selaginella lepidophylla is the scientific name for the plant species, which also has the common name resurrection moss. Explain what is meant by the term species. A group of organisms that can reproduce, or you can say breed or mate, and produce fertile offsprings. So we say, okay, this is one species, this is a cat, this is a dog, this is an oak tree, this is a mango tree. So a group of organisms that can breed and reproduce, and number two, produce offsprings that are fertile. Then part two state the name of the scientific system used to name the species and suggest one advantage of using the scientific name rather than the common name. The name is called binomial nomenclature. An advantage is what? Same name in all parts of the world. It's an international name. Common name can differ in different parts of the world. Genus name helps identify the closely related species. A group of organisms that can reproduce. You can say breed and reproduce, or you can just say reproduce and produce fertile offsprings. That's why horse and donkey are separate species because when they reproduce, they produce a mule which is infertile. So the name of the scientific system used to name the species and suggest one advantage is a binomial system. Got 1 mark for that. And the advantage is same name in all parts of the world. Or you could have said common name can differ in different parts of the world, or you could have said genus name helps to identify closely related species. Now we come to question number 5. Bacillus subtilis is a species of bacterium that is widely used in biotechnology. It can be used to manufacture the protein lipase for industrial use. Figure 5.1 is a diagram of B subtilis. Two of the parts A, B, C, and D. Now what is A? Is the circular DNA. C is the cytoplasm. B is they've shown you the cell membrane, or it looks like there's a cell wall outside it, so it must be the cell membrane, or there can all be a slime capsule as well. And then we have D is the ribosomes. So two of the parts labeled A, B, C, and D in figure 5.1 are involved in the manufacture of proteins such as lipase. Identify the two parts, name them, and explain their role. So letter A, you have to give the letter. So A is the DNA. You can also say genetic material. And what is the function of A? It contains the gene for the protein lipase and codes for the production of the protein lipase. Then the other letter is D. And what is that? That is a ribosome. And what happens on the ribosome? It adds amino acids together to build up the protein. The DNA dictates the sequence of amino acids which are going to be put together to make that protein. So letter A, DNA, letter D, ribosome. DNA contains the gene for the protein, or you can say codes for the production of the protein, or you can say codes for the sequence of amino acids in the protein, or the sequence of amino acids in the lipase. Part D is the ribosome. What does the ribosome do? It adds amino acids together to build. We say it is the site of protein synthesis. That means amino acids are put together here. They come in the right sequence, and then the peptide bonds form between them, and a polypeptide chain is formed. And then the folding is, of course, then automatic. Then it says suggest why B subtilis has flagella. Oh, it can move. Why does it have flagellum? It's motile, or you can say can move. That's all what you have to say. Then it says explain why plasmids are useful in biotechnology. Why? Because we take out these plasmids and then we insert certain genes in it. We put in the gene for insulin. We take out a plasmid, put a gene for insulin, then we put this plasmid, this recombinant plasmid back into a new bacteria. You see, we first destroyed the bacteria when we take out the plasmids. Then we cut the plasmid and we add the gene of interest. Like for instance, we want to put in the DNA which contains the gene for insulin, and we put that into the plasmid and then we join it, and this is called now recombinant DNA. And then we take new bacteria and then we put this into the new bacteria. So they are going to carry the gene, and then this bacteria is going to make insulin for me. So explain why plasmids are useful in biotechnology. So the gene slash DNA can be inserted into them, and then of course we can put this plasmid back into a new bacteria, and then this bacteria is going to make insulin for me, or it can make any desired protein. Like in this case we're talking of the enzyme lipase, so we want the enzyme lipase. So we'll take the lipase gene, we will take plasmids out of bacteria, we'll cut the plasmids, put the gene and plasmid together, and then we will glue it together. We use an enzyme ligase. Now we've got recombinant DNA. So some of it is the old plasmid, and then there is the gene of interest. So we put that here. Now this is called recombinant DNA. And then now we take another bacteria and we put this into another bacteria, and then we culture these bacteria, and these bacteria are going to make insulin for me, or lipase, or whatever gene of interest that I put in it. Now coming to the B part of the question, B subtilis can be grown industrially in large containers. When B subtilis is in ideal conditions, it can reproduce rapidly, increasing the population size and the biomass of bacteria in the container. Graphs E and F. Graphs E and F, figure 5.2, shows how the biomass of B subtilis increases after small populations of bacteria added to two containers with different environmental conditions. Now look at it very carefully. In E, the biomass is increasing. So look at the y-axis. It's in gram per dm cube, and this is the time in hours. So by 8 hours, E is much more and F is lesser. It's only little more than 0.3 and E is about 0.5. Always read off the graphs. Be very careful in reading of the graphs and write things on the graph like E is 0.5 and then F is 0.32. Yeah. So let's now give the name of the large industrial containers used to grow bacteria. That's very simple. Fermenter. Now it says in graph E, the biomass increases as shown in figure 5.2 indicate using the letter M the part of the graph E where bacteria are reproducing most rapidly. So it's here. Because you see a very sharp increase. You see the graph increasing very sharply. And it said part of graph E. Please do not do F. Look at it very carefully. This is the E that we are talking about. Don't get it wrong. Some of you just marked it wrong. Now what does it ask you? So there's 1 mark for that. And it says using the letter M, the part of the graph E. Calculate the percentage increase in mass in the first 6 hours. Now in the first 6 hours, let's look at it in E. It was 0.1 in the beginning. And then it is 0.5. So how are you going to do this working? So from 0.1, what did it say? Percentage increase. So from 0.1, it went up to 0.5. So you minus this, it will be 0.4. And the original was this. So 0.4 divided by 0.1. What is it? Change over original and into 100. Now this is 400%. This is 400%. How do you calculate percentage increase? Is change. So 0.5 minus 0.1 will be 0.4. And the original was 0.1, so it went from 0.1 to 0.5. So change was 0.4 over the original, which is 0.1. And then you multiply it by 100 because you want a percentage change. So everybody must know the change over original into 100. Then coming to part 4, describe two differences between graph E and graph F. Then it says suggest one environmental condition which could have caused the differences between graph E and graph F. Well, it could have been pH, it could have been oxygen, it could have been food supply or nutrient supply, it could have been temperature. Okay, fine. Now let's go back to question part 4. Two differences between E and F. Let's look at E and F now again. Now, if you look at E and F again, I'm going to rub it all off so that you can, very fairly compare it. Now, what is happening? E has a higher final biomass. So E has a final higher biomass. E reaches the maximum biomass quicker. It reaches the maximum biomass here. Here, this one reaches it here. So E reaches the maximum biomass quicker than F. Then E starts at a higher biomass than F here. E starts at a higher biomass than F. And E rate of increase of biomass is faster than F. So rate of increase is faster than F. and then you could give me some figures of it, that it was 0.1 and now it's 0.5. In this case it was much lesser than it, 0.2, 0.6, and then it went up to 0.32. So you give me some figures and that gets you a mark as well. That's always sensible to give me figures for this sort of a question. So E has a higher F final biomass than F. E reaches a maximum biomass quicker. E starts at a higher biomass, and E rate of increase of biomass is faster than F. Then coming to part C of the question. Now coming to the C part of the question, lipase is also produced by the human body. Explain why it is produced and how its function is different from the function of bile. Lipase digests fats, or you can say hydrolyzes fats, or you can say breaks down fats into fatty acids and glycerol, which are produced as a result of the breakdown of fats. While the bile does emulsifies fat, so increases the surface area of the lipids. And also bile neutralizes the acidic food, so providing the optimum pH for digestion. So bile and lipase are two different things. Bile emulsifies the fat, so it breaks the big fat into smaller droplets. Now this makes it easier for the enzyme lipase to now work on it. So this is the enzyme lipase. So it's going to break this down fat quickly. Otherwise it would take a lot of time, because the lipase would have to work here on this large molecule, on this large globule. It's not a molecule, sorry, it's a large globule. So lipase digests fats into fatty acids and glycerol. Bile emulsifies fats or increases the surface area of the lipids, and bile neutralizes the acidic food. You only have to give me 3 points out of these. There are actually 4 mark scheme points and a maximum of 3, so any 3 of these. Question number 6 is the last question. A beam of bright light shines into a human eye for 2 seconds. A similar beam of light shines onto one side of the tip of a plant stem for several days. Describe the ways in which the human and the plant coordinate their responses to these beams of light. Your answer should include stimulus detection and method of responding. So in the humans, it is detected by the retina. It involves electrical impulses. Response is muscular contraction in the iris and causing a constriction or reduction in the size of the pupil. In plants, it is detected by the stem tip. Involves hormones auxin, which move or diffuse to the side away from light, causing more growth on the shaded side, and this is called positive phototropism, because the plant stem is going to grow towards the light. So I'm just writing it on a separate page, but you would of course write it here on the paper. You won't write it on a separate page. So in the humans, it is detected by the retina, which has light receptors, and this involves neurons or the nervous system and carries electrical impulses. Generates electrical impulses. Then the response is muscular contraction in the iris, and this causes constriction or reduction in the size of the pupil. You could have said constriction if you know that word. If you didn't know that word, you could have said reduction in the size of the pupil. In plants, it is the light is detected by the stem tip. This involves hormones, production of hormones / auxin. Auxin diffuses to the side away from light, causing more growth on the shaded side. And this is called positive phototropism, in which the plant stem is going to grow towards the light. So this is the tip. You're giving light from this side. Now, what is going to happen is the auxin which is produced is going to be on the outer side. So this is going to cause more growth. It's going to be more growth. It won't bend. So this is going to grow towards light. I do not draw a diagram like this and then I say, oh, well, this one is going to go. No. It's not the same stem. It's going to be further growth. That is not going to bend. So please understand this. Then coming to the last part of the question, B part, describe the role of synapses in a reflex arc. What is the role of synapses in a reflex arc? Transmission of impulse between neurons. In one direction, a neurotransmitter is released into the synaptic gap. This causes, then it results in a diffusion across the gap. and then it binds with the receptor protein. So, this is all a very direct question. Describe the role of synapses in a reflex arc, transmission of impulse between neurons, one direction, neurotransmitter released into synaptic gap, diffuses across the gap, and then finally binds with receptor protein. This completes this paper. Thank you very much for watching, and please do leave your comments if there's anything else that you need which I can help you with your exam preparation. Thank you very much.